BioArchive, the preprint server for biology, audio recording, read by author Jessie M.N.G. Lopez. A genetic screen for suppressors of cryptic 5' splicing in C. elegans reveals roles for KIN17 and PRCC in maintaining both 5' and 3' splice site identity. Authors Jessie M.N.G. Lopez, Kenneth Osterhout, Tatiana Holland Cartwright, Saul Katzman, Alan M. Zoller. This article is a preprint and has not been certified by peer review. Author summary. Pre-mRNA splicing is an essential step of gene regulation carried out by an unusual molecular machine, the spliceosome. Unlike other molecular machines, such as ribosomes that simply assemble and catalyze chemical reactions, the spliceosome is a highly dynamic cycle carried out by 5 specialized small nuclear RNAs and over 100 proteins, which sequentially join, rearrange, and withdraw from the splicing assembly during each splicing cycle. These assemblies initially choose splice sites where the pre-mRNA will be cut, and then undergo multiple rearrangements to finally form the active site, which catalyzes the splicing reactions, which remove an intron from a pre-mRNA. We are currently in the midst of a resolution revolution with ever clearer cryo-EM snapshots of stalled complexes, allowing researchers to visualize moments in time in the splicing cycle. These models are illuminating, but do not always elucidate mechanistic functioning. Therefore, our lab takes a complementary approach, using the power of genetics in a multicellular animal to gain functional insights into the spliceosome. Using a C. elegans genetic screen, we have found novel functional splicing roles for 2 proteins, KIN17 and PRCC. Our results suggest that the spliceosome does not just rely on its initial identification of the splice site, but in a later step, re-identifies where to cut. We liken this 2-stage identification to using a microscope by first using the coarse focus to find the area of interest, and then using the fine focus to adjust as needed. This work moves us closer to a full mechanistic understanding of how the spliceosome chooses where to cut a pre-mRNA message. Competing interest statement. Authors have declared no competing interest. Abstract. Pre-mRNA splicing is an essential step of eukaryotic gene expression, carried out by a series of dynamic macromolecular protein RNA complexes, known collectively and individually as the spliceosome. This series of spliceosomal complexes define, assemble on, and catalyze the removal of introns. Molecular model snapshots of intermediates in the process have been created from cryo-EM data. However, many aspects of the dynamic changes that occur in the spliceosome are not fully understood. Caenorhabditis elegans follow the GU-AG rule of splicing, with almost all introns beginning with 5' GU and ending with 3' AG. These splice sites are identified early in the splicing cycle, but as the cycle progresses and the custody of the pre-mRNA splice sites is passed from factor to factor as the catalytic site is built, the mechanism by which splice site identity is maintained or reestablished through these dynamic changes is unclear. We performed a genetic screen in C. elegans for factors that are capable of changing 5' splice site choice. We report that KIN17 and PRCC are involved in splice site choice, the first functional splicing role proposed for either of these proteins. Previously identified suppressors of cryptic 5' splicing promote distal cryptic GU sites. However, mutations in KIN17 and PRCC instead promote usage of an unusual proximal 5' splice site, which defines an intron beginning with UU, separated by just one nucleotide from a GU donor. We performed high-throughput mRNA sequencing analysis and found that mutations in PRCC, but not KIN17, changed 5' splice sites genome-wide, promoting usage of nearby non-consensus sites. We further found that mutations in KIN17 and PRCC changed dozens of 3' splice sites, promoting non-consensus sites upstream of canonical splice sites. Our work has uncovered both fine and coarse mechanisms by which the spliceosome maintains splice site identity during the complex assembly process. Introduction. The spliceosome is not one distinct machine, but a series of dynamic macromolecular protein RNA complexes that assemble on and catalyze the removal of introns from pre-mRNA transcripts in eukaryotic organisms. Over 100 proteins, including multiple helicases and the 5 U-rich small nuclear RNAs, snRNAs, join, rearrange, and withdraw from a spliceosomal complex in a choreographed sequence over the course of a single splicing cycle, catalyzing the removal of an intron and ligation of flanking exons, references 1 and 2. Spliceosomes assemble de novo from subunits on each nascent pre-mRNA intron. Multiple spliceosomes often interact with a pre-mRNA transcript at the same time, and different introns in a pre-mRNA can have different kinetics for removal. Reference 3. The splicing process is responsible for an essential information processing step in the flow of genetic information, and almost all protein coding transcripts in metazoans must be spliced in order to become functional. Early in the metazoan splicing cycle, 3 important landmarks on the nascent pre-mRNA are identified by spliceosomal components: the 5' splice site, the exon-intron boundary, the branch point, and the 3' splice site, the intron-exon boundary. The U1 snRNA has a 9-base sequence. From 3' to 5', it reads G, U, C, C, A, Psi, Ca, Ua, which pairs with the bases of the 5' splice site, reference 4. A perfectly complementary 5' splice site would have the sequence from 5' to 3', CAG/GUAA/GUAU, where the slash represents the splice site. However, this exact sequence is rarely found at verified 5' splice sites in metazoans. Instead, the consensus sequence that has some overall base pairing ability with U1 snRNA, with a strong preference for a /GU dinucleotide to start the intron, is what's seen. Reference 5. That first G is nearly invariant. Its 5' phosphate will link directly to the branch point adenosine. For the 3' splice site, the U2AF heterodimer initially identifies the polypyrimidine tract and AG dinucleotide at the end of the intron. U2AF65 binds the polypyrimidine tract, and U2AF35 binds the nearly invariant AG/ at the very 3' end of the intron. Reference 6. U2AF helps recruit U2 SNRP to the branch site, where base pairing interactions with U2 snRNA, in which the branch point adenosine is bulged out of the duplexed region, define the branch point. References 6 and 7. Mutations in splice sites or in cis-regulatory regions, such as enhancer or silencer binding sites, can cause a variety of deleterious splicing phenotypes that are associated with disease phenotypes. Examples include exon skipping, intron inclusion, and frameshift mutations. Mutation of a splicing donor or acceptor sequence leads to the activation of nearby cryptic splice sites, which are defined as splice sites that are functional but activated only when an authentic splice site is disrupted by mutation. In the human gene mutation database, about 9% of inherited disease-causing mutations alter splice site sequences, reference 8, and another about 25% of disease-causing mutations affect splicing by disrupting other important sequences, such as nearby binding sites, references 9 and 10. Some aberrant mRNAs are degraded by nonstop or nonsense-mediated decay pathways, so that the possibly toxic effects of aberrant mRNAs are not amplified into many aberrant proteins by polyribosomes, reference 11. Precise splicing is central to gene expression, and mutations that affect splicing can lead to a variety of deleterious phenotypes. Throughout the many dynamic assembly steps of the splicing cycle, the U1 identified 5' splice site is maintained by a series of protein and snRNA escorts. In the earliest steps of the spliceosome assembly, the 5' splice site is directly bound by U1 snRNA, reference 12. In the transition from pre-B to B complex, U1 leaves the spliceosome while handing the 5' splice site off to U6 and residues of PRP8, references 13 and 14. From B complex, the spliceosome undergoes a number of rearrangements through pre-B Act 1, pre-B Act 2, B Act, and C complex. Cryo-EM studies of these complexes from the human spliceosome, references 2 and 15, allow for the study of different snapshots of the spliceosome assembly process. In these complexes, there is an exchange of different factors that interact with the region of the 5' splice site and its interactions with U6, Akagaga box, as the 5' splice site is loaded into the catalytic core of the splicing machine. Proteins and SNRPs that bind the 5' splice site must bind precisely to a degenerate sequence on a long nucleotide chain, maintain their exact binding position through helicase-powered translocations and substantial conformational changes, and then transfer custody of the 5' splice site to the next escort without introducing positional error. It is still unclear which components of the spliceosome ensure that the handoffs between escorts will not result in small shifts in the 5' splice site definition. Thanks to the researchers fueling the ongoing cryo-EM resolution revolution, we now have structures of spliceosomes at many time points in the splicing cycle. These snapshots of experimentally stalled spliceosome assemblies offer valuable insights into the complex assembly pathways, rearrangements, and interactions of spliceosomal components. Mass spec experiments and chemical probing of structures have provided additional information about where and when specific components are associated with the spliceosome during the splicing cycle. These advances continue to build towards a fuller picture of the many multi-step assembly pathways of the splicing cycle and then the organized dissolution of the complex. While the structuralists reveal which proteins are where, geneticists are positioned to provide complementary insights into the functional roles of splicing components in a splice site choice. Our lab has previously made use of an unusual 5' splice site mutation in C. elegans as a tool to reveal residues on splicing proteins that can contribute to splice site choice, references 16 and 17. UNC73 is a guanine nucleotide exchange factor that's important in axon guidance and other aspects of C. elegans development. A fortuitous G to U mutation of the first nucleotide of the 16th intron of the UNC73 gene, allele E936, reference 18, converts the nearly invariant GU dinucleotide found at the beginning of eukaryotic introns to a UU dinucleotide, creating a curiously ambiguous splice site, figure 1A. This splice site mutation results in missplicing, causing the uncoordinated or UNC phenotype, reference 19. This dramatic phenotype is corrected by even a small increase in in-frame splicing, making its suppression screenable. Previously identified dominant mutations that are able to suppress the UNC phenotype by altering cryptic splicing in the UNC73 E936 were found in U1 snRNA, reference 20, SNRP 27, reference 16, and 21, and the largest and most conserved protein in the spliceosome, PRP8, reference 17. The suppressive role these mutations play in the splice site assay provided genetic evidence of a role for these protein residues in 5' splice site choice. After publishing these data, the progress made in cryo-EM and crystal structures of the spliceosome has allowed these suppressor alleles to be precisely mapped in the high-resolution inner core of spliceosomal structures. These mutations are often modeled near the active site of the spliceosome, providing some clues as to mechanisms for maintaining the identity of the 5' splice site during spliceosome assembly. Here we report new additional suppressor alleles identified in the UNC73 E936 genetic screen for suppression of uncoordination that have a dramatically different mechanism of suppression through splicing. Previous suppressors promoted the use of both the -1 and wild-type cryptic splice sites, separated by one nucleotide, the /G/UU, over a downstream cryptic GU splice site donor at position +23. Here, we identify two new proteins as splicing factors in which mutations promote use of the UU splice donor over the adjacent GU splice site. Two missense alleles in the worm homolog of KIN17, kinship to REC-A, called DXBP1 downstream of XBOX in C. elegans, and an overlapping point mutation and deletion in the worm homolog of human PRCC, proline-rich coiled coil protein or papillary renal cell carcinoma protein called PRCC1 in C. elegans, promote the usage of an unusual UU splice site in 3-choice, 2-choice, and 2x2 choice cryptic splice site assays. High-throughput mRNA-seq studies reveal that these mutations affect global splicing at native splice sites. But despite similarities in effects on UNC73 E936 cryptic splicing, mutations in KIN17 and PRCC display strong but very different effects on native genes. These results are the first demonstration that PRCC and KIN17 have roles in maintaining splice site identity during spliceosome assembly. Results. Mutations in KIN17 and PRCC alter cryptic 5' splice site choice in UNC73 E936. The UNC73 E936 allele has a G-to-U mutation at the first nucleotide, that's the +1 of the 16th intron. This mutation presents the spliceosome with an ambiguous 5' splice site, resulting in usage of two out-of-frame cryptic 5' splice sites and a striking uncoordinated phenotype. Reference 19, figure 1a. The majority of the splicing occurs at a GU dinucleotide found 23 nucleotides into the intron, the +23 site, resulting in an out-of-frame message. An additional 12% of splicing occurs at a position 1 nucleotide upstream of the wild-type splice site, the -1 site, using the new GU dinucleotide formed by the E936 mutation, also resulting in an out-of-frame message. These out-of-frame messages are not substrates for nonsense-mediated decay, reference 19. An additional 13% of splicing occurs at the wild-type splice site, even though this defines an intron that begins with a non-canonical UU. Only the small fraction of splicing at the in-frame UU splice site produces full-length functional protein. The animals bearing the UNC73 E936 allele are able to live and reproduce through self-fertilization, but are profoundly uncoordinated. Even a modest increase in splicing at the in-frame UU splice site results in a dramatic phenotypic reversal, which is visible at the plate level, making this allele a sensitive assay of perturbations to splice site choice. References 16, 17, and 19. Because those previous screens have identified mutations on residues modeled near the active site of the spliceosome, and those mutations often change global 5' splice site choice, we concluded that a genetic screen using this allele can identify loci which are capable of affecting splice site choice. Because we have never found the same mutation twice In 500,000 mutagenized genomes screened previously, we hypothesized the screen is not yet saturated. Therefore, we performed the genetic screen again to search for more suppressor alleles and splicing factors capable of altering splice site choice. In a recent iteration of the E936 extragenic suppressor screen, we recovered 4 new extragenic suppressor alleles with improved locomotion and a novel change in cryptic splicing. Using Cy3 labeled primers in reverse transcription polymerase chain reaction, RT-PCR, visualized after denaturing gel electrophoresis, we found that these 4 strains displayed a different pattern of cryptic 5' splice site usage in UNC73 compared to wild type, but, curiously, also a different pattern compared to previously identified modifiers, references 16, 17, and 19. While previous suppressors have reduced splicing at the +23 site with coordinated gains at both the -1 and wild type sites, these 4 new suppressors had the most dramatic effect in altering the relative usage of the -1 and wild type sites relative to each other, resulting in increased wild type splice site usage to around 25% of UNC73 messages, consistent with the improved locomotion phenotype identified in the screen, figure 1b. We now refer to extragenic suppressors in 3 classes. U1, type 1 is the U1 snRNA suppressor, SUP39, while type 2 includes the protein factor suppressor alleles SNRP27, M141T, and PRP8, P524S, and G654E. The type 1 and type 2 suppressors both reduce +23 splice donor usage with concomitant increases in both the -1 and wild type splice sites. The dramatic change in the relative -1 and wild type usage is the key feature of these new type 3 suppressors. The 4 new type 3 suppressor alleles capable of changing 5' splice site choice are in the C. elegans homologs of KIN17 and PRCC. Using Hawaiian strain SNP mapping, reference 26, as described in methods, we mapped each of these 4 new suppressor alleles to an arm of a chromosome. Then, using high-throughput DNA sequencing of the strain genomes, we followed by SNP identification protocols to identify differences in genomic sequence from the starting UNC73 E936 uncoordinated strain in methods. We identified spliceosome-associated proteins and RNA binding proteins with mutations in their sequence within the chromosomal delimited region. The 4 new type 3 suppressor alleles capable of changing 5' splice site choice are in the C. elegans homologs of KIN17 and PRCC. Using Hawaiian strain SNP mapping, reference 26, as described in methods, we mapped each of these 4 new suppressor alleles to an arm of a chromosome. Then, using high-throughput DNA sequencing of the strain genomes, followed by SNP identification protocols to identify differences in genomic sequence from the starting UNC73 E936 uncoordinated strain, see methods, we identified spliceosome-associated proteins and RNA binding proteins with mutations in their sequence within the delimited chromosomal region. 2 of the suppressor alleles had point mutations in a gene, DXBP1, the worm homolog of KIN17, a mutation that changes the 23rd amino acid from a lysine to an arginine, K23N, AZ105, figure 1b, and another that changes the 107th amino acid from a methionine to an isoleucine, M107I, AZ33, figure 1b, lane 4. Both of these residues are conserved between worm, human, yeast, and Arabidopsis, figure 2. C. elegans DXBP1, downstream of Xbox binding protein, or DOX1, is the homologue of a human and mouse gene known as KIN, or KIN17. It is not a kinase. Except in the multiple sequence alignment, figure 2, throughout this manuscript, we will refer to KIN17 when talking about the protein and DXBP1 when talking about the gene. The 23rd residue of the worm homologue of KIN17 is proximal to the CHC2 zinc finger in a region predicted to be near the U6 pre-mRNA helix in BAC2. References 1524 and figure 3. The 107th residue of the worm homologue of KIN17 resides on a 310 helix in a loop in the atypical winged helix domain. This domain is atypical because the cluster of residues that are typically positively charged and coordinate nucleic acid binding in a winged helix is not charged, leading to the hypothesis that the highly conserved 310 helix is involved in protein binding, reference 24. KIN17 is predicted to have a disordered central region flanked by alpha helices, reference 15, followed by a tandem of SH3-like domains separated by a flexible linker. KIN17 was first identified in a search for mammalian homologues of the bacterial DNA repair protein REC-A, and has since been studied primarily for roles in DNA damage repair and transcription in eukaryotic cells, references 27 through 37, or cancer, references 38 and 39. In S. cerevisiae, there's a gene named RTS2 that shares homology with the N-terminal portion of KIN17, reference 40. Observations about KIN17 include the following. KIN17 binds to single-stranded and double-stranded DNA, references 37, 41 through 45, with a preference for AT-rich curved double-stranded DNA, references 31, 46, and 47. Binds to RNA with domains exhibiting preferences for specific polynucleic acid oligos, references 48 and 49. KIN17 also binds to proteins in complexes of high molecular weight, including ones involving chromatin, references 41, 45, and 50, DNA recombination, reference 46, DNA damage repair, reference 51, DNA replication, reference 36 and 44, pre-mRNA splicing, reference 48, 52 to 55, and 15, and translation, reference 45. It is likely that KIN17 performs more than one role in the eukaryotic cell. This screen also identified 2 mutations in PRCC1, the worm homologue of human PRCC. A mutation which changes the 371st amino acid from an isoleucine to a phenylalanine, I371F, AZ120, figure 4, and a large deletion near the C terminus that removes amino acids 298 through 377 in frame, AZ103, figure 4. Except in the multiple sequence alignment, throughout this manuscript, we will refer to PRCC when talking about the protein and PRCC1 when talking about the gene. PRCC, known variously as proline-rich protein, proline-rich coiled coil, papillary renal cell carcinoma translocation-associated gene protein, and mitotic checkpoint factor protein, has been implicated in oncogenic fusions where the proline-rich N-terminal region is fused to any of several transcription factors, references 56 to 58. The proline-rich region is relatively proline poor in C. elegans compared to human. The domain is absent in Arabidopsis. The 371st amino acid of the worm homologue of PRCC occurs in the middle of the longest stretch of identity, where 9 residues are conserved from worm to human. The deletion suppressor identified in the screen overlays that region, figure 4. PRCC has been identified as a potential spliceosomal B-act complex component by mass spec, reference 59, and yeast 2 hybrid, reference 60. To confirm that the 3 single amino acid substitution alleles are indeed responsible for the altered cryptic splicing of UNC73 E936, we used CRISPR-Cas9 to generate the same amino acid substitutions from scratch, see methods, and tested these programmed alleles for an effect on the ratio of -1 to wild-type splice site usage. The CRISPR-generated PRCC1 AZ102 allele can suppress UNC73 E936 splicing and movement defects, confirming the identity of PRCC I371F suppressor, figure 1C, lane 5. A deletion null allele of PRCC1 generated by the C. elegans Gene Knockout Consortium, GK5556, is viable and can both suppress the movement defects of UNC73 E936 and alter cryptic splice site usage, figure 1C, lane 6. This demonstrates that PRCC is a non-essential gene and that loss of function leads to changes in splicing. Confirmation of the DXBP1 alleles by CRISPR is a little more challenging, as they map to the same chromosome as UNC73, making crosses difficult. And injection of CRISPR-Cas9 RMP complexes into E936 animals is challenging, as the worms are sick and have smaller brood sizes. We solved this challenge by generating the 2 DXBP1 mutation alleles by CRISPR in a wild-type strain, followed by a subsequent CRISPR mutation of an UNC73 to mimic the E936 allele. These strains resulted in suppression of UNC73 uncoordination and the predicted change in -1 to wild-type splice site usage, figure 1C, lanes 3 and 4. To understand whether KIN17 is an essential gene, we used our standard CRISPR pipeline to generate a DXBP1 null allele, see methods. We put the DXBP1 null allele over a fluorescent HT2 balancer designed such that homozygous DXBP1 positive animals are GFBP positive, but homozygous lethal. Heterozygous animals are GFP positive, and animals homozygous for DXBP1 null do not fluoresce. We found that KIN17 deletion is embryonic lethal in C. elegans. Occasionally, GFP animals homozygous for DXBP1 null can survive to something resembling L3 stage. However, these rare animals are severely underdeveloped and do not live to molt again. Simultaneously, the C. elegans deletion mutant consortium, reference 61, created a DXBP null allele and also found the deletion of DXBP1 to be homozygous lethal. KIN17 and PRCC promote usage of a non-canonical UU 5' splice site in 2-choice and 2x2 choice reporters. We were interested in the unique suppressive phenotype displayed by mutations in KIN17 and PRCC, so similar to each other, but distinct from previously identified suppressor phenotypes, in that they change the relative 5' splice site usage of overlapping GU and UU splice sites. In order to investigate this further, we utilized an intragenic suppressor allele of UNC73 E936 AZ30, in which an A to G mutation at the 26th intron eliminates the usage of the +23 cryptic splice site, figure 5A. Therefore, the only 2 splice sites available are the cryptic GU and the non-canonical UU, 1 nucleotide downstream. We refer to it as the 2-choice substrate. In the wild-type background, these 2 splice sites are used about 41% and 59% of the time, respectively, figure 5B, lane 3. In a KIN17 K23N, KIN17 M107I, or PRCC I371F background, we see altered ratios of splice site choice in the 2-choice splice site competition assay relative to the wild-type background, figure 5b. The splicing pattern was similar in the presence or absence of the plus 23 GU splice site. Compare with figure 1c. Despite the GU being the primary hallmark of the 5' splicing landmark, these suppressor alleles are promoting usage of an adjacent UU 5' splice site. In the KIN17 K23N, KIN17 M107I, and PRCC I371F strains, the relative UU splice site usage is increased to 77%, 67%, and 76% respectively, figures 5b and 5c. In the two-choice splice site competition assay, we found that mutations in PRCC and KIN17 promote usage of a non-canonical UU splice donor over an adjacent upstream GU splice site. We wondered whether the information to promote this UU splicing was contained within the 5' splice site itself, or whether it was promoted by some nearby splicing enhancer element, or whether it was dependent on a distance from the original splice site. To answer these questions, we devised a new competition assay which would separate sequence from location. Using CRISPR-Cas9 and a repair oligo, the region bearing the curious GUU 5' splice site doublet was duplicated in the native UNC73 gene and inserted downstream, overwriting the downstream bases of the intron, figure 5a, allele AZ100. This doubled the splice donor doublet, creating the 2x2 choice splice site assay, featuring the two splice site choice doublets 18 bases away from each other. We knew the second doublet was close enough to be chosen by the spliceosome because it was proximal to the plus 23 site from the three-choice splice site assay in the original UNC73 E936 allele. We abolished the plus 23 site so that only the four choices contained in the two doublets remained. In a wild-type background, both splice sites of the original doublet are used more than either of the splice sites in the duplicated doublet downstream. In the upstream doublet, there's a slight preference for the UU splice site, 53%, while the less used downstream doublet, the UU site is less preferred, 34%. Figure 5D, lane 3. When this doubled doublet, UNC73 AZ100 allele, is combined with suppressor alleles, KIN17 K23N, KIN17 M107I, or PRCC I371F, we see altered ratios of splice site use in the doubled doublet 2x2 choice splice site competition assay relative to wild type, figure 5D. In all three cases, both doublets are used and most splicing comes from the upstream doublet. In the presence of any of these three suppressor alleles, the usage of the UU splice site increases relative to the GU splice site in both the original doublet and the duplicated doublet 18 nucleotides downstream. When the ratio of splice site usage at each doublet is considered independently, for KIN17 K23N, KIN17 M107I, and PRCC I371F, we see that for both doublets, usage of the UU splice site is increased, figure 5e. These data support the hypothesis that the information for switching to the UU splice donor usage in the presence of these three suppressor alleles is dependent on the 5' splice site sequence and not a distance from some other marker on the pre-mRNA. Analysis of splicing changes in native genes in the presence of KIN17 and PRCC suppressor alleles. Because mutations in KIN17 and PRCC are able to promote usage of 5' UU splice sites in our splice site competition assays, we wanted to know if these mutations also change splice site choice at native loci. The UNC73 transcript upon which all of our splice site competition assays are built is not subject to nonsense-mediated decay, reference 19, which is why we are able to recover cryptically spliced frame-shifted transcripts. However, when looking for alterations displaying splice site choice more broadly, we expect that most transcripts will be targeted by nonsense-mediated decay, NMD, especially given that the prominent splicing change we might expect to see would move the start site of an intron over by a single nucleotide, thus changing the reading frame. Given that many out-of-frame messages would be targeted by NMD, it might be difficult to detect these changes in splicing, as they may potentially lead to differential transcript stability. C. elegans is a rare metazoan that is able to survive without a functional NMD pathway, making it possible to do the experiment in an NMD knockout background, reference 62. We designed a CRISPR-Cas9 engineered SMUG4 null allele, AZ152, which is easily detectable by single worm PCR and restriction digest, allowing for ease of mapping and crosses. SMUG4 was chosen for creating an NMD strain as it is not located on the same chromosome as either DXBP1 or PRCC1. We confirmed that the new SMUG allele is NMD defective by both the presence of the protruding vulva phenotype and the accumulation of NMD-targeted isoforms of RPL12, data not shown, reference 63. We use genetic crosses to create strains with KIN17 K23N, KIN17 M107I, PRCC I371F, or PRCC null, combined with SMUG4 AZ152, isolated mRNA, and performed mRNA-seq on 3 biological replicates for each suppressor strain, as well as the original SMUG4 AZ152 mutant strain as a control. So 15 libraries in total. We performed 75 by 75 base pair paired end reads and obtained between 46 million and 69 million reads for each library. We performed star mapping, which we modified to accommodate UU 5' splice sites, as described in methods. We ran an alternative splicing analysis, which looked at both annotated and unannotated alternative 5' and 3' splicing events, as well as ensemble annotated skipped exon, mutually exclusive exon, multiply skipped exon, intron inclusion, alternative first exon, and alternative last exon events. For each alternative splicing event, we quantified relative usage of each junction in each of the 15 libraries. We then compared the percent spliced in, PSI, for each event between each library and the starting SMUG4 mutant strain. We performed pairwise comparisons between each of the 3 biological replicates of the suppressor strain against each of the 3 biological replicants of the NMD mutant strain alone, for a total of 9 comparisons for each alternative splicing events, and asked how many of those 9 comparisons generated a delta PSI of over 15%. Those events for which all 9 pairwise comparisons had a delta PSI of over 15%, that's the pair sum 9, were then analyzed individually on the UCSC genome browser with the RNA-seq tracks, reference 64, to confirm the alternative splicing events. We then filtered those confirmed pair sum 9 events for those where there was an over 20% average delta PSI in the 9 pairwise comparisons. Table 1 summarizes the number of confirmed alternative splicing events meeting these criteria in each strain comparison. PRCC I371F and PRCC null promote usage of 5' UU splice sites and degenerate 3' GU splice sites throughout the C. elegans transcriptome. Using the stringent criteria described above, we were able to identify multiple examples of changes to 5' splicing in the presence of PRCC mutations. In PRCC I371F and PRCC null, we found respectively 34 and 46 examples of introns where mutant strains promote usage of a downstream UU splice site over an adjacent GU splice site, figure 6b. This type of intron start of GUU 5' splice sites is similar to the UNC73 E936 splice site choice in competition assays. Many of the introns affected by PRCC I371F were also affected by PRCC null, figure 6e. These introns are enriched for an A in the fourth position of the intron immediately following the invariant GUU, figure 6b. In PRCC I371F and PRCC null backgrounds, we also found 37 and 44 instances, respectively, of events where the alternative 5' splice site promoted in the presence of PRCC mutations were at GU dinucleotides, either 2, 3, or 4 nucleotides away from the wild type GU dinucleotide. Most of these shifted downstream, figure 6e. A substantial portion of the introns affected by the PRCC null were also affected by the point mutation in PRCC I371F, figure 6d. Surprisingly, despite the similarities in splicing phenotypes observed in our UNC73 E936 based splice site competition assays for both PRCC and KIN17 mutations, we found negligible examples of changes to 5' splice site choice at endogenous introns in the presence of either of the two KIN17 mutant alleles. PRCC1 null affects longer introns, especially in the case of non-GUU introns. We were interested in the group of introns affected by PRCC mutations, so we looked at the lengths of introns and flanking exons. Despite the overlap of affected introns, the average intron length for each group is very different. Because rare, very long introns can exert a strong influence on averages, we report median intron length. In order to focus more on the relative contribution to median intron length in each category, we removed events in common and looked at the lengths of introns unique to each dataset, figure 6d. While the median intron length for UU and GU alternative splice sites promoted in PRCC I371F background is similar to the overall median intron length in C. elegans of 51 nucleotides, reference 25. The median intron length of PRCC null promoted alternative introns for both UU and GU introns is much longer, with a median intron length of 320 and 552 nucleotides respectively, figure 6F. KIN17, K23N, KIN17, M107I, and PRCC null promote usage of weak upstream 3' splice sites throughout C. elegans transcriptome. Even more surprising than the inability of KIN17 mutations to change global 5' splice site choice is the ability of those same mutations, identified in a screen for modifiers of 5' splice site choice, to affect 3' splice site choice. The 3' splice sites promoted in the presence of these KIN17 mutations were highly degenerate sites, figure 7a, mostly located 6 or 7 base pairs away and unidirectionally upstream of the adjacent consensus 3' splice sites, figure 7b. We found 108 examples of alternative 3' splice site usage in KIN17 K23N, 23 examples in KIN17 M107I, and 35 examples in the PRCC null, table 1. We only found one example of 3' splice site changes in PRCC I371F. Most of the intron events identified in KIN17 M107I were also represented in KIN17 K23N events, figure 7c. We only found 5 unique examples of PRCC null mutations affecting 3' splice site choice that are not shared with the KIN17 mutant strains. The unidirectional shift to a poor consensus stream upstream 3' splice site is similar to the developmentally regulated alternative splicing events in which cells in the C. elegans germline show more splicing to an upstream poor consensus alternative 3' splice site relative to somatic cells, reference 25. In that study, 203 alternative 3' splice site events were identified as being developmentally regulated. 49 of those alternative 3' splicing events overlap with the alternative 3' splicing events identified in PRCC and KIN17 mutants. Figure 7D. Discussion. This work represents the first direct demonstration that KIN17 and PRCC have a role in splice site choice. Prior to this manuscript, KIN17 was classified in the spliceosome database under miscellaneous proteins found irregularly with the spliceosome, and had been primarily studied for roles in DNA damage repair and cancer, not splicing. We report here that mutations in the N-terminal unstructured region, K23N, and in the winged helix, M107I, of KIN17 promote usage of an unusual UU 5' splice site, mostly downstream of an adjacent GU splice site, figures 1 and 6. And even more surprisingly, these same mutations change 3' splice site choice at over 100 native loci, promoting degenerate splice sites upstream of canonical 3' splice sites, figure 7. Excitingly, while we were preparing this manuscript, a structure of pre-BAC2 spliceosome was published, reference 15, with KIN17 modeled in this transient intermediate near what will become the active site later on in the splicing cycle, figure 3. The loop and 310 turn of the winged helix are positioned facing the active site, though the M107 residue points into the globular core of the winged helix, not outward. This leads us to hypothesize that the M107I mutation repositions nearby outward-facing residues, such as the highly conserved nearby aromatic residues, histidine 107, histidine 106, and tryptophan 112. Townsend et al. Hypothesize an early transient role in spliceosome assembly for KIN17, proposing that KIN17 prevents components of the spliceosome, including PRP8 and BER2, from prematurely entering the BAC conformation. In light of our results showing significant alterations to 3' splice site, we hypothesize that KIN17 either has an additional later role in the splicing cycle, or that the premature assembly of BACT leads to later acceptance of an upstream branch point corresponding to a degenerate 3' splice site, as the branch point itself is not yet positioned for catalysis in BACT. This demonstration of KIN17 as a bona fide splicing factor may potentially point to a closer association between pre-mRNA splicing and DNA damage repair than is currently understood. PRP19 is a multifunctional ubiquitin ligase known to be a component of both spliceosomal and DNA damage repair complexes, reference 65. And a recent study showed that U1 SNRP and components of the DNA damage response compete for binding at human 5' splice sites, reference 66. As both splicing and DNA damage repair require the recognition, cutting, and joining of nucleic acid chains, it may not be too surprising that they share some factors in common. Prior to our studies, PRCC had a firmer association to the spliceosome, identified as a factor in BACT complexes through yeast-2 hybrid and mass spec experiments, references 13 and 60. But no functional role had been identified, nor had it been modeled into any metazoan spliceosomal structures. There's no S. cerevisiae homologue of this factor. We report that the I371F point mutation, located in the 9 residue long region in the C terminus of PRCC that is identical between worms and humans, changes 5' splice site and native loci, and PRCC null promotes both non-canonical downstream 5' splice sites and non-canonical upstream 3' splice sites. It is possible that PRCC is serving a different function in C. elegans than it does in other organisms. The proline-rich region of PRCC, most often found in oncogenic fusions, is noticeably proline-poor in the C. elegans homologue relative to humans. The identification of a suppressor point mutation in a conserved region of the C terminus points to a potential key region for splicing control. The discovery of this new class of suppressors of UNC73 E936 cryptic splicing has led us to think about the splice site like a piece of evidence in a criminal case, held by escorts, which shuttle the precise genetic landmarks through dramatic conformational changes. Each escort of the 5' splice site must by nature hold it reversibly. Therefore, slipping or disengagement are possible while the 5' splice site is in the custody of a SNRP guardian or other protein factor guardian, especially when the pre-mRNA is under tension from helicases or other components of the spliceosome. If we follow the chain of custody, we expect that translocations and changes of possession are likely to be the inflection points where alterations to splice site identity relative to the initial identification by early factors are more likely. Some factors capable of affecting splice site choice may assist during those vulnerable moments in the splicing cycle. When an escort repositions or lets go entirely, these factors may make nucleotide shifts less likely. We see in the presence of the suppressors identified in the study that the spliceosomal components are choosing degenerate splice sites. The positions we have identified in KIN17 and PRCC may serve to prevent such slips in wild type during vulnerable points in the chain of custody. These mutations display a different splicing phenotype from previously identified suppressors. Instead of the predictable reduction of the distal +23 site and relatively even increase in usage of both splice sites of the doublet observed in factors previously identified, figure 1D references 16 and 17. This new class of type 3 suppressors displays a sharp change in the ratio of usage of the two adjacent splice sites of the doublet, with the downstream UU site promoted over the adjacent GU site, figure 1D. This effect is seen with or without other nearby cryptic 5' splice GU splice sites, figure 1 and figure 5B, and can be replicated at a downstream location, figure 5D. We believe this difference between class 3 suppressors and previously identified suppressors supports the idea that these factors act at a different point in the splicing cycle. The first U1 dependent step of 5' splice site identification can be thought of like the coarse focus on a microscope, and the class 2 suppressors can be thought of as mutations that maintain the general region of the identified splicing target. In later steps, after U1 has left, we can think of the maintenance of the 5' splice site as more of fine focus function, perhaps related to U6 identification of the 5' splice site, reference 67, and the class 3 suppressors are mutations that alter the ability of the spliceosome to maintain the fine focus of the splice site that will be used in chemistry, an effect that is consistent with a duplicated doublet switching results, 5D. We were surprised that this genetic screen for factors that affect 5' splice site choice identified factors capable of affecting both 5' and 3' splice site choice. We were further surprised that despite the similar splicing phenotypes displayed in the UNC73-based reporters of splice site choice, there were differences in how suppressors affected global splicing in an NMD background. Both PRCC suppressors affected global 5' splice site choice, promoting usage of non-consensus 5' splice sites downstream of canonical 5' splice sites, especially along introns, but neither of the two KIN17 suppressors affected global 5' splicing. Both KIN17 suppressors affected global 3' splice site choice, promoting usage of non-consensus 3' splice sites upstream of canonical 3' splice sites, as did PRCC null, but not PRCC I371F. No suppressors identified in the screen promoted significant numbers of other splicing alterations, such as alternative first or last exons, exon or intron inclusion or skipping, table 1. All effects observed were local, usually shifting 5' splice site choice by 1 or 2 nucleotides downstream, and 3' splice sites choice either by 6 or 9 nucleotides upstream. This preference for upstream non-consensus 3' splice site reminded us of the tissue-specific 3' splice site switching identified by our lab, reference 25. In addition, many of the upstream AGs found to be preferred in germline tissue relative to somatic tissue were also preferred in KIN17 and PRCC mutants relative to wild type, figure 7D. Mutations in various parts of the spliceosome act on the same splice sites, this work and unpublished data. A simple interpretation of this overlap is there are a relatively small number of ambiguous 3' splice sites present in the genome, and that there are multiple mechanisms involved in making that distinction. Our work shows that the genetic probing of subtle changes to splice site choice, by which we have studied various 5' splice site mechanisms, can also be used to study 3' splice site choice mechanisms. Despite these mutations affecting the choice made during the second splicing reaction, we shouldn't take these results as strong evidence that KIN17 and PRCC are still present and functioning in late spliceosomal complexes. PRCC and KIN17 may be influencing the choice before the second step occurs. One possibility is that these mutations alter branch point choice, and then the alternate 3' splice site choice is a secondary effect. Another possibility is that these mutations alter the form of the spliceosome to affect 3' splice site choice in a way that persists during the second step, even after the protein themselves are removed. Methods. Full step-by-step protocols of many of the methods described below have been deposited at protocols.io. Growth conditions. C. elegans were maintained at 20 degrees Celsius on nematode growth medium NMG agar plates inoculated with OP50 E. coli. Strains were discovered in the suppressor screen, genetically engineered using CRISPR mutagenesis, created by doing genetic crosses, or were obtained from the C. elegans Gene Knockout Consortium, reference 61. Mutagenesis and identification of putative suppressed strains. Age-synchronized, uncoordinated UNC73E936 hermaphrodites in gametogenesis, larval stage L4, were soaked in 5 micromolar N-nitroso N-ethylurea, ENU, as previously described, reference 16. After extensive washing, 4 animals were placed at the edge of an OP50 E. coli seeded 10 centimeter NMG agar plate for 500 plates and allowed to self-propagate. NMG plates were maintained at 20 degrees. Whereas the UNC73E936 animals' movement defects confined them in place, after 8 days, suppressed F2 animals were able to crawl away from the crowded pile of uncoordinated animals and are identifiable by their improved locomotion on the far side of the plate. Identification of extragenic splicing suppressors. The UNC73 gene is suppressed from this screen. The UNC73 gene in suppressed lines from the screen was sequenced to distinguish between extragenic and intragenic suppressors. One of the intragenic suppressors, UNC73E936AZ30, is used in this study, figure 5A. Remaining extragenic suppressor alleles were mapped to chromosomes using a strategy described in references 26 and 69. Briefly, each suppressor strain identified in the genetic screen was crossed against a polymorphic Hawaiian isolate, CB4856, and uncoordinated F2 animals that continued to have Only uncoordinated offspring were recovered. These new UNC strains were then screened for regions that are homozygous for SNP markers as described by reference 26. Approximately 20 uncoordinated strains for each exogenic suppressor strain outcrossed to the Hawaiian strain were recovered and DNA extracted and combined. For each chromosomal region, we expected to see a mix of Hawaiian and Bristol N2 single nucleotide polymorphism, SNPs, except in the region linked to the suppressor mutation, where we expect to see 100% Hawaiian SNPs, loss of suppressor in the N2 background. And in the region of UNC73, where we expect to see 100% N2 SNPs, the uncoordination allele is in the N2 background. Using this approach, we will able to narrow down the suppressors to approximately 1/3 of the length of the chromosome. At the same time, the suppressor strains were backcrossed 2 times to the N2 wild-type strain, and then we performed high-throughput genomic sequencing of the suppressor strains. We used STAR, reference 70, to map those sequences back to the C. elegans genome. Diploid SNPs relative to the original N2 strain were identified using GATK, reference 71. The SNP-F tool, reference 72, was used to identify SNPs within genes in the chromosomal region identified in Hawaiian strain mapping. That list of putative suppressors was cross-referenced to the Eureka Lab spliceosome database, reference 73. And candidate spliceosome-associated genes and RNA binding proteins in the delimited genomic regions were chosen for further analysis. The suppressor allele identity was verified by de novo recreation of each putative suppressor allele using CRISPR-Cas9 genome editing, and those resulting in both suppression of the movement defect and molecular changes in splicing were identified as bona fide suppressors. CRISPR-Cas9 genome editing. Cas9 guides were chosen from the CRISPR guide track on the UCSC genome browser. C. elegans reference assembly, WS220-CE10, references 64, 74, and 75. And cRNAs were synthesized by Integrated DNA Technologies, IDT DNA. Cas9 CRISPR RNA guides were assembled with a standard tracer RNA. These RNAs were heated to 95 degrees C and incubated at room temperature to allow joining. The full guides were then incubated with Cas9 protein to allow for assembly of the CRISPR RNA complex, reference 76. That mix, along with a single-stranded repair guide oligonucleotide, was then micro-injected into the syncytial gonad of a young adult hermaphrodite animals. A dumpy 10 CN64 co-CRISPR strategy was used to identify F1 animals showing homologous recombination CRISPR repair in their genomes, reference 77. Silent restriction sites were incorporated into repair designs so that mutations could be easily tracked by restriction digestion of PCR products from DNA extracted from single worms. Injected animals were moved to a plate in recovery buffer, reference 76, allowed to recover for 4 hours, and moving worms were then plated individually. F1 offspring were screened for the dumpy 10 CN64 dominant roll co-injection marker phenotype. F1 roll animals were plated individually, allowed to lay eggs, and then the adult was removed and checked for allele of interest by PCR, followed by restriction enzyme digestion and gel electrophoresis. If an F1 worm showed the presence of a heterozygous DNA fragment matching the programmed restriction site, non-rollers in the F2 generation of that worm were screened by electrophoresis of digested PCR products. Individuals that had lost the co-injection marker but were homozygous for the allele of interest were retained and sequenced at the gene of interest to verify error-free insertion of sequences guided by the repair oligo. RNA extraction, cDNA production, and PCR amplification. RNA from indicated strains was extracted from mixed stage populations of animals using trizol reagent from Invitrogen, then alcohol precipitated. Total RNA was reverse transcribed with gene-specific primers using superscript 3, Thermo Fisher, or AMV reverse transcriptase, Promega. cDNA was PCR amplified for 25 cycles with 5' Cy3 labeled reverse primers, IDT, and an unlabeled forward primers using either Taq polymerase or fusion high fidelity polypyrimidase from New England Biolab. PCR products were separated on 40 centimeter tall, 6% polyacrylamide denaturing gels, and then visualized using a molecular dynamics Typhoon scanner. Band intensity was performed using ImageJ software. RNA-seq. Triplicate total RNA isolations were done for each strain, and mRNA sequencing libraries were prepared for each RNA isolation by RealSeq Biosequences, Santa Cruz, California. 75 by 75 paired end reads were obtained on a NovaSeq 6000 sequencer with 9 libraries combined in a lane. RNA-seq results were trimmed, subjected to quality control, and 2 paths aligned to the UC Genome Browser C. elegans reference assembly WS220CE10. This earlier assembly release was used to facilitate comparison to previous RNA-seq datasets obtained by our lab. Using a modified version of STAR, reference 70. The standard version of STAR, in addition to the canonical GU-AG intron motifs, supports GC, AG, and AU-AC motifs for the 5' and 3' splice sites. Because C. elegans does not have minor spliceosomes with AU at the 5' end of its introns, we modified the STAR source code to use UUAG as the third motif in place of AUAC. Furthermore, we ran STAR with parameters that adjusted the default score gap ATAC, effectively score gap UUAG in our modified version of STAR, junction penalty from -8 to 0, so that the program would treat UUAG spliced introns with the same scoring as GUAG introns. High stringency delta PSI analysis. Alternative 5', alternative 3' events found on the STAR mappings of all libraries were identified and filtered for those introns with at least 5 reads of support, total across all samples, and a maximum of 50 nucleotides between the alternative ends, either 5' or 3' respectively. In addition, alternative first exon, AF, and alternative last exon, AL, skipped exon, SE, retained intron, RI, mutually exclusive exon, MX, and multiple skipped exon, MS, events were derived from the ensemble gene predictions archive 65 of WS220CE10 using junction counts prefer pairwise events function. The percent spliced in, PSI, in each sample was derived for all of these events using junction counts. Pairwise differences in PSI between samples for the above events were calculated. Alternative splicing with a minimum 15% delta PSI were included for further consideration. Each strain had 3 biological replicates. Therefore, between any 2 strains, a total of 9 pairwise comparisons were possible between each suppressor strain and the SC340 SMUG4 comparison strain for each alternative splicing event. For each suppressor strain, only alternative splicing events that showed a change in the same direction, more than 15% delta PSI compared to the SMUG4 control in all 9 pairwise comparisons, pair sum equal 9, were considered. Those events in which the mean delta PSI over 20% across all 9 comparisons were included for further consideration. The reads supporting that alternative splice site choice event were then examined by eye on the UCSC genome browser reference assembly to ensure that the algorithmically flagged events actually looked like real examples of alternative splice site choice. Supplemental Table 1 has the chromosomal location, PSI measurements, and notes for all alternative splicing events that fit those criteria. Sequencing data access. Raw mRNA sequencing data for 15 libraries in FASTQ format, along with the .gtf files for all analyzed alternative splicing events, are available in FASTQ format at the NCBI Gene Expression Omnibus, GEO. Consensus motifs. Consensus motifs were created using web logo, reference 78. Multiple sequence alignments. Multiple sequence alignments were generated using the EMBL EBI Clustal Omega MSA tool, reference 79. Acknowledgements. We thank Noel Eng and Amy Castellanos for technical assistance. We are grateful to Michael Duty, Melissa Jerica, Manny Aries, Harry Noller, and Jordan Eisinga for helpful discussions. We thank Josh Arabear for assistance identifying mutant alleles from sequencing data, and Guillaume Chemfro for the suggestion of using Phi3 labeled PCR primers. Research in the Zoller Lab was supported by a grant from the National Science Foundation and currently by a grant from the National Institutes of Health. JMNGL was supported by UCSC MCD Graduate Training Grant. Author contributions: Jessie MNG Lopez, conceptualization, formal analysis, investigation, methodology, project administration, validation, visualization, writing, original draft preparation, writing, review, and editing, and audiobook. Kenneth Osterhout, investigation, methodology, writing, review, and editing. Katiana Holland Cartwright, formal analysis, investigation, validation, visualization. Saul Katzman, data curation, investigation, software, writing, editing, and reviewing. Alan M. Zoller, conceptualization, funding acquisition, investigation, methodology, product administration, resources, supervision, writing, review and editing, and validation. Thank you for listening.